Our next speaker this morning is Dr. Ken Ramos. He is the Distinguished University Professor of Biochemistry and Molecular Biology, the Director of the Center for Environmental Genomics and Integrated Biology at the University of Louisville. And he's going to talk to us today about a story for environmental influences and the role of epigenetics on lines and reprogramming of the mammalian genome. Ken, do you want the lavalier mic or the podium mic?
Well, this is a tight clip. This one? Okay, good. Okay, that last question, I think, really set the way for much of what I have to talk about. So I thank you for that and the comments that John made. I also want to thank the organizing committee, Thea, Cheryl, for the invitation. It's always a great pleasure to see friends and tell them about the work that we do. And, of course, it's always great to listen to John talk about L1s. His work actually has inspired much of what we've done in this field. So for that, I'm grateful, as well as for some of the reagents that he's made available to us as we have ventured into this story. What I will do today is basically put into context much of what you heard from John for the environmental health sciences and recapitulate some of the key points that he made in the context of L1 biology, but more important than that, kind of take it beyond that into how do environmental chemicals modulate these processes and what are the biological implications of that, those events. One of the things that we will talk about this morning actually relates to the ability of L1 to interact and hijack sort of the biochemical machinery of cells to kind of do what it does. So the work will focus on mobilization just a little bit. You've heard lots about that already. It will primarily target the concept of L1 as a key regulator of epigenetic and chromatin dynamics in cells. Which I actually think is one of the major reasons that cells have engineered to keep L1 for as long as they have. And that's, I think, one of the reasons that makes L1 so important. The last thing that I will tell you about is the role of L1 in reprogramming the genome, which I think is in the context of genome plasticity, which is the talk, the theme of the meeting. Many of you probably have seen this figure. This came from Science when they basically wrote a eulogy of the junk DNA. This just came out about a couple of weeks ago and sort of followed the slew of papers that came from the ENCODE project, which is celebrating its 10-year anniversary. I think there's some important things that basically have come out from ENCODE. From my perspective, the most important one of which is the fact that what we thought was junk obviously is not junk. That includes, line 1 for the most part. And the idea that what we called junk in the past might actually serve regulatory functions in the genome to actually provide templates that encode instructions for the genome to do what it does. And I actually think that that is one of the major biological functions that L1 has retained. Major technological advances over the past 20 years have really made possible what we do today. I remember 20 years ago when I first made the observation that environmental carcinogens activated L1, you went to the literature and you found nothing regarding this topic. I mean, really a handful of studies. The first time that I did a search on this topic, I literally only identified 40 papers that I could touch, many of which had no relevance to mammalian biology. So we've come, I think, a long ways, and the important lesson that I think has been hidden in what we call junk DNA is the fact that much of that codification actually is related to chromatin structure and chromatin dynamics. Big surprise that coding genes really don't hold the answer for many of the questions that we've asked ourselves. And then the question is, what is this extra DNA doing that is so important to the regulation of cell function? And I believe that that is at the level of regulation. I think when you talk about jumping genes, you always have to talk about Barbara McClintock's work. Certainly very controversial in the field of genetics when she first put forth the ideas of jumping genes, and the concept that DNA was actually not static and that it could actually move around. We've since obviously come up with the term of junk. Now junk is no longer junk, and today we will be talking about retrotransposons and repeat sequences. Much of the difficulty, the technological challenge that we faced over the years in looking at retrotransposons is because of the repetitive nature and the fact that the assays just were not very friendly and amenable to use them. You're now familiar with this cartoon, so I don't have to spend too much time telling you about it. Just to remind you that the enzymatic activities which are encoded by this full-length L1 include that ORF1, which is a ribonucleoprotein that's very important in the assembly of that complex that John made reference to, and the activities coming off of the second open reading frame, endonuclease and reverse transcriptase, which are important in carrying out the mobilization functions of L1. I think I do want to point out that despite the fact that we call L1 junk, you must recognize that this looks like a gene and smells like a gene and functions like a gene, but that in addition to that, it actually carries functions which I think extend beyond that. And I'll tell you a little bit about that in a minute. John covered, I think, in excellent detail some of the molecular consequences of L1 mobilization. I will not tell you much about that. The only idea that I want to sort of seed in your mind so that when you look at the data, you actually can react to it, is the fact that perhaps we need to rethink the biology of L1 and recognize that the majority of what L1 does, given the fact that cells have engineered mechanisms to repress L1 mobilization, might actually lie elsewhere and not just be mobilization per se, but actually be functions of L1 that might be unrelated to mobilization. That's just an idea that I want to seed for you. With that in context, there is the question of does L1 function to reprogram the genome? If it does, which of course I believe it does, and I have some data to support that idea, is that reprogramming a function of mobilization alone, or does it involve functions of L1 which are unrelated or maybe related but not tightly linked to mobilization? And then for me, since I'm interested in human disease and the connection between L1 and human pathogenesis, the question becomes, does this mobilization of L1 or do the other functions of L1 account for either risk for disease onset or changing of disease patterns and progression in individuals? And I think there's a lot to be found and learned from that perspective. So why is it that we don't have to worry as much as what Cheryl, I think, was alluding to in her question to John? Cells have been smart enough to realize that the hijacking that can take place when L1s are mobilized could potentially be dangerous to the genome. So what they've done is they've engineered at every step of the way, which is related to the question that was being asked at the end of John's talk. Every step of the way, cells have engineered mechanisms that will repress and control L1 biology. That level of redundancy, I think, has served us well. It basically ensures that when you go and drink a little bit of alcohol, your genes don't start moving around. It ensures that when you have to write a grant, at 2 o'clock in the morning and then get up to give a talk the next day, hopefully your L1 is not moving around. And that level of control, despite the fact that can be exercised at every step of the way along the mobilization cascade of L1, primarily relies on epigenetic control. It is silencing through DNA methylation that basically keeps L1 quiet. And we've spent a lot of time trying to understand mechanisms of L1 reactivation by environmental carcinogens. And the example that I will give you today relates to benzoate pyrene. A few words about mechanisms of L1 silencing. I've already mentioned hypermethylation of the L1 promoter. There's quite a few CpG islands in this promoter. We've actually studied all of those in the human promoter and found that only 2 appear to be important to the regulation of Bence-Paring-induced reactivation. A very important concept that I think Kazazian advanced a few years ago is the idea that some of the truncated RNAs generated in either direction of the L1 promoter might actually function as silencing RNAs for cells, not only for L1 itself, if the process is aborted, before completion of the transcription elongation process takes place, but also for other target RNAs. And I think there's a lot of interesting biology that probably will emerge out of this. One point that John made was the fact that the L1 template is an unfriendly template. And in fact, there's a significant elongation deficiency with the polymerase falling off of the promoter. Hello? John, yes, John?
Quick question. So those silencing RNAs, do they arise by random accidents and then they just happen to function as silencing RNAs and have to function before get observed?
We don't know very much about how they come about. Some of that origination actually stems from the fact that the polymerase, as it's walking the template of the L1, falls off, and then it generates this double-stranded, this single-stranded and double-stranded RNAs that basically are targets for dicer and become processed as RNAs. So 5' UTR truncation, which obviously kills the L1. Repressor proteins, we talked about ApoBec, and the work that I will show you today focuses on RB, an aryl hydrocarbon receptor, which is a receptor protein that many of you recognize in the field of environmental health sciences. It turns out that both RB as well as AHR function in silencing RNA. And of course, this has huge implications in the context of some of the cell stochastic paradigms that Thea mentioned yesterday, for molecular switches that control major attractor modes in cells, and of course it has importance in the context of environmental control, since aryl hydrocarbon receptor is a protein that is recognized by many environmental chemicals. I think in the reading assignments or reading suggestions that we gave you, I cited this particular article, so some of you might have actually seen some of this. But in 2009, we published our first study looking at mechanisms of control of L1 expression. It turns out that what the evidence at that time showed is that much of the repression of L1 and the major mechanism by which L1 is regulated involves the ability of RB, retinoblastoma, to recognize specific sites in the 5' untranslated region of full-length L1s, to assemble a repressor complex through protein-protein interactions that inhibit the ability of the promoter to fire. And that macromolecular assembly involves basically recruitment of histone deacetylases as well as enzymes which modulate the covalent modifications of histones for opening of chromatin and firing of the promoter. Turns out that RB is recruited to both the human and the mouse, and I think there's some important architectural differences that we want to talk about, and I'll make reference to that in a few minutes. The ability of RB to be recruited to that promoter is actually a function of E2F binding to that promoter. So the L1 promoter contains E2F binding sites. Those binding sites actually accessorize the process through interactions with RB for assembly of that complex, which is repressive to L1 transcription. The binding of E2F to the promoter is actually RB independent. And of course, the question remains whether E2F, through interaction with other transcription factors, might actually regulate the function of L1. And importantly, we did not see recruitment to human endogenous retroviruses, which, as John pointed out, are primarily dead sequences, speaking to some specificity. The bulk of the work that we did to characterize the regulation of line 1 through an RB-mediated mechanism relied on a deletion model generated at Louisville, in which we generated RB knockout cells that were deficient for all forms of the RB family of proteins. And so using this genetic tool, we were able to examine and discretize interactions of RB with line 1 that might actually be important in the regulation of patterns of gene expression. In RB null cells, if in fact RB is important in the regulation of L1, what we found was that L1 levels were greatly enhanced in cells as you knock down in a dose-dependent manner RB levels. So that basically there's an endogenous role for retinoblastoma proteins in regulating the levels of expression of line 1. And of course that opens questions, important questions in terms of what happens in the context of an RB mutant phenotype and what happens in the context of environmental exposures that change the way in which RB interacts with other proteins and basically hijacks RB away from lines and probably allows for unregulated expression of mobile elements. The reactivation of line 1 in the function of the RB null phenotype appears to be primarily a function of recruitment or lack of recruitment of the histone deacetylases as shown by chip assays, as well as modification in the level of DNA methylation. Now, one thing that I wanted to make sure that I pointed out is that when you look at reactivation profiles for L1, whether that is in the context of benzpyrene reactivation or whether that is in the context of RB null phenotypes, what you find is that the kinetics of line 1 expression are actually very complex and that they show a triphasic pattern of regulation, which actually appears to match directly the biochemical events which are taking place in the reactivation process. And I'll explain what that means. If you were to do a time sequence measurements of L1 in the context of an L1 reactivation, what you find is that there's waves of induction of the L1 that track to some of the biochemical changes which are taking place as a function of progression into the reactivation state. The early phase is an induction phase where basically within 8 hours of reactivation, you see modification of chromatin structure through alterations in histone deacetylase recruitment to promoter sites of the L1. Then about 90 hours after the reactivation process, you see another phase of induction of the L1 message, which parallels DNA methylation profile in cells, suggesting that in fact there is crosstalk between the chromatin dynamic changes which are taking place through histone deacetylase macromolecular complex disassembly and the DNA methylation machinery, and this crosstalk has actually been documented in the literature, and then a stabilization of messenger RNA levels. I'm making this point because I think this tracks very nicely with some of the comments that Thea was making yesterday regarding attractor states in cells and how cells have fluidity in their genetic expression such that they can transition from one state to the other, and I think this is applicable to L1. Importantly, if you were to pharmacologically inhibit HDACs, you actually increase L1 expression. So here is the first set of data in the context of environmental control. I made reference to the first set of experiments that we did where we found that chemical carcinogens capable of causing extensive DNA damage, and this relates to the question that Cheryl was asking John, reactivate line 1. And then the question is, why would this be? Is it that the chemical is interfering with biochemical processes involved in the repression, silencing of L1? Or is it that in the face of DNA damage, cells sense the need to reactivate line 1 as a function of DNA repair, and we don't actually know exactly what the answer to that is, but certainly have continued to work on it. One of the first set of experiments that we did, and this dates back to the early 90s, were studies in which we then try to track and understand the reactivation process by benzoapyrene, a chemical carcinogen which is found in basically every form of organic combustion pollution that you can think of. Consider a prototypical polycyclic aromatic hydrocarbon, a chemical carcinogen that basically works by causing single point mutations in GC-rich target genes. This parent molecule, in order to exert carcinogenic effect, requires metabolic activation through cytochrome P450s to generate a number of reactive intermediates that actually are the ones that carry toxicity for the chemical. Turns out that in the context of benzoapyrene, you see dose-dependent reactivation of L1, and this is a northern blot for L1. Those of you who are familiar with northern blot, since this is almost extinct technology now, probably recognize that there's multiple bands here which actually speak to that processivity issue that I mentioned before, where the polymerase falls off and generation of truncated fragments as a function of processing. Up here you actually have full-length transcript. And an important finding that we made at that time was the fact that metabolites of benzoapyrene, which were capable of causing DNA damage and or inducing oxidative stress in cells, actually reactivated the L1 better than the parent molecule, speaking to the idea that, in fact, the reactivation process required that metabolic component for complete reactivation. We went on at that time to show that the reactivation that you see by benzoapyrene as well as these oxidative metabolites in the mouse, which is different from the human, was actually a function of reactive oxygen species regulatory elements that were found within the L1 promoter that gave rise to activation and regulation of that promoter. So that the first piece of evidence that we accumulated at that time was that, at least in the mouse, oxidative stress appeared to be one of the mechanisms by which L1 could be reactivated. And I think this actually opens an important biology in the context of some' the complexity of the biological networks that L1 regulates, and I'll tell you about that in a few minutes. Here is the architecture of the mouse 5' UTR compared to the human. John made reference to this being an internal promoter. Suffice it to say for now that you've got an E2F binding site here. Other important transcription factors that have been shown to primarily repress transcription of L1 include STRI and RONX3 as well as YY1. We could talk about those during the question and answer period. So it turns out that there is transcriptional control to L1, and that transcriptional control actually involves either oxidative stress in the mouse or regulation and assembly of complexes that regulate the promoter.
It looks like the major difference that you're seeing is in processivity, because those shorter truncated transcripts aren't really changing much, but you're getting a big change now in whether or not the full length is present.
Absolutely. Absolutely. So taking advantage of the beautiful system that was engineered by John in '96, to look at retrotransposition, we basically asked ourselves the question, okay, is the reactivation of L1 through interference with RB inhibition of the L1 transcription as well as oxidative stress associated with mobilization in cells? And of course, at that time, we were primarily interested in seeing if BAP and BAP-related carcinogens were actually able to complete the full cycle of L1 retrotransposition. And so utilizing this assay, we based in HeLa cells, we actually showed that, lo and behold, yes, benzoapyrene as well as BAP-related carcinogens are able to reactivate the line 1 that have been silenced, and that this reactivation was actually capable of completing full cycles of retrotransposition in mammalian cells. Utilizing the same assay, we've actually gone on to show that this mobilization, John, is in response to one of the questions that you raised, actually does take place in non-transformed cells. We've seen reactivation and full cycles of retrotransposition in smooth muscle cells. And we've also seen it in embryonic kidney cells quite efficiently, actually.
But you're using fetal cells, doesn't it imply that it's going to be RB independent?
Actually, that's a very good point.
Repeat the question for the
Somebody asked whether the use of HeLa, because of the RB status, actually impacts the process. And what it does basically makes it a more permissive system for retrotransposition to take place. But that relates to the question that I was trying to discuss with John, which is that non-RB mutant states also allow for retrotransposition to occur. Okay, so here's a cartoon that captures, I think, some of what I've told you, because I want to move to discussions of the reprogramming. Basically, in the face of benzoate pyrene injury, you've got a promoter that it's silenced through RB interactions. Benzoate pyrene interferes with the ability of RB to keep this promoter in the repressed state. And once this chromatin opens, you create a permissive environment that, upon cell division, actually allows for changes in DNA methylation. This transition states depicted in this slide, I think, parallel the kinetics of L1 induction, which I made reference to in the context of early induction of the RNA message, which is accounted for chromatin remodeling, and later stage upregulation of L1 message, which appears to be accounted for by changes in the level of methylation. One thing that I did not discuss with you because of time is the fact that a key disruption exercised by benzoate pyrene in this system is the degradation through protease ubiquitin pathways of DNA methyltransferases, primarily DNA methyltransferase 1, not the inducible forms, that allow for downstream alterations in the pattern of DNA methylation of the L1. And what appears to be emerging in the context of this story is that the reason that BAP-type carcinogens are so effective in reactivating line 1 is because of the sustained reactivation that appears to be taking place. So this is not a quick response that basically fades away and that probably allows for the repressor mechanisms to jump in and halt the process, but that is sustained for an extended period of time in a way that allows for processing of the complete mobilization cycle. And we can talk about that more as the talk progresses. Yes? Yes.
So putting this in the context of the processivity, the change in DNA methylation would suggest a change in initiation of transcription as opposed to transcription elongation, which is what you were really seeing.
No, remember that the processivity deficit relates to the ability of the polymerase to walk that template. The deficits in elongation really come from the unfriendly nature of that template, with the polymerase basically falling off of it. Not related to DNA methylation. What I'm talking about is that the transcriptional activation of L1 has two components to it. One component that actually involves changes in the covalent modification status of the histones that allows for unwinding of the chromatin template, and changes in the DNA methylation status of that promoter that allow for continuous firing of the promoter. And this is all in the span of probably 100 hours worth of time. So there is that proteasomal dependent degradation of DNA methyltransferases. Reference to the reactivation. Turns out that when the RB repressor mechanism is lost in this promoter, there is now opportunity for activating marks in the DNA to appear that allow for unwinding of the template. And, of course, we actually confirmed that much of what BAP does must involve changes in the DNA methylation status by using silencing RNA technology to basically mimic the ability of DNMT1 silencing to recapitulate much of what benzoate pyrene was doing. And then the last part of what I want to talk to you about relates to what are the biological consequences of this reactivation. So I've shown you some evidence that chemical carcinogens can in fact reactivate and overcome silencing mechanisms engineered by cells to keep the L1 quiet. And the question then that I'd like to challenge you with is twofold. Is there a consequence to that reactivation process, number one? And number two, does the reactivation require mobilization for the reprogramming of genes? Up until not very long ago, much of what we understood about L1 biology assumed that what L1 did was related to mobilization, so that you could do all the list, of things that John presented to you, as well as the listing that I gave you. Insertional mutations, duplications, inversions, rearrangements, so forth. And that is important. I'm not saying that it's not. But the question is there more to what L1 is doing? And I believe that there is. Now, one thing that is going to be important for you to remember is that the changes in chromatin dynamics associated with the reactivation might actually be important to the reprogramming process itself. So this slide actually captures what's driven my research for the past 30 years. Basically, the whole idea of genome plasticity, the whole idea that cells can transition. I probably should have put, arrows going back in the other direction. Cells can, in fact, transition and are plastic in moving from one phenotypic state to another. And that perhaps what environmental cues are doing is making the transitions from one state to the next doable. And in so doing, you have to rethink what we think of when we think of toxicity. People think of toxicity as basically the cell's dead, right? But perhaps that's not it, especially when you think of this in the context of low-dose exposures. When you think of this in the context of the exposures that we face. Those exposures, to the work of many toxicologists, no longer kill you because we can identify up front what is damaging to you. But what might be happening is that low-dose exposures might actually be tweaking with the molecular machinery of cells, L1 being an example, to actually reprogram what cells are encoded to do. And I will show you some example of that. Focusing on L1 as one of the key nodes in regulatory pathways that might actually be driving that. When high-throughput technologies came about, I was one of the first people to jump in the bandwagon. Mainly because I've always was puzzled and troubled by the idea that one gene at a time was not going to give us the answer if you were trying to understand genomic plasticity, if you were trying to understand transition states in terms of phenotypic control. And so we took advantage of high-throughput technology to construct what I call a genetic regulatory network for line 1. The thinking here was there was very little that we understood about what L1 did in cells. What proteins do L1 proteins interact with? What processes get hijacked as a function of the mobilization? What interference is taking place? And so we constructed this genetic regulatory network, and the beauty of that genetic regulatory network was that it was constructed in the complete absence of any biological knowledge. Okay? So it was truly a computationally derived network, and I can tell you the details of the construction of the network. And the reason that I wanted to do it that way was because I didn't want to bias what little we knew about the interactions of L1 proteins with other proteins within the cell with what I wanted to find out. So I wanted to basically go for unknown. And this is what we came up with. Two things I think were really interesting in that biological network. The first one was that RNA polymerase II, which is the polymerase that is utilized in processing of L1 transcripts, was a key node in the network. And the second point was that PAS proteins actually emerge as a major node in the network. And Pas proteins is a superfamily of which aryl hydrocarbon receptor belongs to. And of course, that to me raised important questions. We then went on to pursue what role Pas proteins might actually have in the regulation of line 1, and I've already alluded to the fact that AHR is actually essential for the reactivation process that takes place as a function of carcinogen treatment, and that ligands for the aryl hydrocarbon receptor through AHR receptor mediated mechanisms actually regulate L1 transcription. But what about some of the other elements within the network? And so we started experiments to try to reconstruct the biology of the network. So we basically did a reverse engineering process. We started with no biological knowledge, and then I started piling bits of information in terms of what might actually be taking place. The first set of experiments that we did was an experiment in which we focused on AHR as a node within the L1 regulatory network and basically silenced L1 and asked the question, do other members of the genetic regulatory network change as a function of this manipulation? And the answer to that is yes. If you change one of the nodes, you actually change members which are unrelated to that node. So there is connectivity to the network. And we then decided, okay, how are we going to study and discretize mechanisms by which L1 might be regulating the expression of these genes? So these are genes which are not directly interacting with L1. What is the connection between the two? So what we did was we turned back to the same conceptual paradigm that John had developed, but we changed it a little bit in that we now inserted tags for both ORF1 and ORF2 that we could utilize to track the expression of this construct, not just for selection, but the expression of these proteins, and identify interacting partners for these proteins through this process. And so we've actually taken advantage of the same cassette that we were using before, inserted strep A here and a make flag tag here. And now we can actually track the expression of these proteins in cells as a function of the reactivation process. And of course, we're utilizing now this system to be able to track the interaction with other elements in that genetic regulatory network. In keeping, I think, with what we understand about L1 biology, notice that there is marked abundance of the ORF1 protein, ORF2 not quite so much, which I think is what others in the field have shown. And I think another important observation is that the localization of these proteins, at least under unstimulated conditions, is primarily a cytoplasmic localization with some minor localization inside of the nucleus of cells. We also took advantage of the point mutation that John alluded to that kills reverse transcriptase activity in the construct, so that we can start asking the question, if there is reprogramming of the genome, if that reprogramming involves the genes which I've identified in the non-biological regulatory network, is this mediated through mobilization or not? So now we can localize these proteins and basically ask that question. This is unpublished data. The paper is actually ready for submission. I was kind of tweaking with it yesterday, hoping that we can get it out. So I'm pleased to share it with you. It's a complicated story, not too surprising. Turns out that you can actually cluster all of the elements within that predictive regulatory network that I showed you into these clusters that I've shown you here. Okay? That's the first observation. Second observation, speaking to the power of computational prediction in biology, a network which we constructed 5 years ago in the absence of biological knowledge gave us 92% predictive value for the elements which were predicted as high predictors of L1 biology in cells. I was astounded by that finding. And the third important observation that came is that just as we had suspected, not everything that L1 does requires mobilization. So in the context of changing programs in cells, in the context of instructing the cellular machinery to do what it does to modulate phenotypes, maybe there is biology that's unrelated to the mobilization process. And the reason that we know that is because you could look at the number of genes that changed or did not change in the absence of reverse transcriptase activity, and confirmation that in the absence of reverse transcriptase activity, you did not have complete cycles of mobilization, because we actually did metaphase spreads to localize L1 insertions in cells, utilizing the tags that we had built into the constructs. So that we now have a way of discriminating, if you would, targets of L1 that might be dysregulated during the course of cell progression through transition states, through mobilization mechanisms, relative to targets that might be dysregulated through other mechanisms. And I'll tell you more about what those other mechanisms might be. For now, I want to move on to share with you some other thoughts. The only thing that I will say is that we immediately became very interested in VCAM1 because of the strength of the association with L1 RT-dependent mechanisms, and have actually shown that L1 actually co-localizes with VCAM1 in this network, which I think is going to be very interesting for the regulation process. Those experiments that I showed you were done in the HepG2 cell background, and HepG2 cells were chosen because this is a model that's extensively utilized, and actually is part of the ENCODE set of family of cell lines which are being used. The question that we asked, because when we discretize those elements in the regulatory network, 3 biological processes emerge as important. Adhesion, and I made reference to VCAM1, inflammation, which parallels the oxidative stress concepts that I was showing you early on in the talk, and metabolic control of cells. So we started asking ourselves questions related, does this mean then that when you reprogram the genome in the face of an L1 reactivation, which of course in this case we did it ectopically through insertion of constructs that are highly active, then does that translate into phenotypic changes in cells? And the answer is yes. It turns out that HepG2 cells, which is an epithelial type cell, actually becomes mesenchymal-like in nature when you overexpress L1. And the transition towards a mesenchymal phenotype, i.e. the genomic plasticity afforded by the overexpression of L1, does not require mobilization. So, mechanistic insights that I want to leave you with. The defining attribute of L1s, of course, is their ability to remodel the genome landscape. One food for thought for you is the idea that remodeling of the landscape is not strictly limited by the insertion at a new location and the disruption of whatever structure there might be there, but that perhaps because L1s are so critical in heterochromatin formation, the presence or the absence of L1 might actually be regulating chromatin states where they are located at, such that when you reactivate, when you open up that template, not only are you opening up the L1 template, but you are now allowing for recruitment of regulators of other genes downstream, upstream, elsewhere. And I think the real sort of beauty of what this data seems to be opening up is the idea that L1 might actually be a master regulator of chromatin states in cells. That molecular machinery for silencing and reactivation is subject to environmental interference, something that's going to be very critical for us to try to decipher, because if we're trying to understand how environment facilitates transitions from healthy states into disease states, we're going to have to then embrace the idea that perhaps this reprogramming might actually be part of the way in which chemicals are doing it. We're just beginning to scratch a surface in terms of what these chemicals might actually be influencing in the system. I think something that's really interesting and provocative is the idea of this genetic-epigenetic intersection. For 70 years now, we have thought that carcinogens like benzoate-pyrene worked by causing point mutations in target genes, right? That's what we've learned for all this time. Maybe we need to rethink that. Maybe what we need to start thinking about is that many of these carcinogens and the old paradigm of initiation, promotion, progression might actually have to fall through the crack, and that we're going to have to start thinking of cancer, not just thinking of disorders of growth and differentiation in the context of genomic plasticity. And the idea that perhaps what you're doing is that you're instructing the cell to behave in a different way. And perhaps L1 is one of the ways in which this is happening. And I think that pathogenesis might actually involve that disruption. I'll skip this one since I've already talked about it. I will only make reference to the fact that there's a whole bunch of conditions that have been associated with L1 insertions. Now we're going to have to worry about perhaps some disorders that might actually involve non-insertional mechanisms. And I think the beauty of it is that now we have ways in which we can target the epigenetic machinery of cells to see if, according to the Thea's principle of reversibility, the way in which we can manage disease now is through mechanisms that modulate the reprogramming that took place to damage the problem to begin with. And I'd like to close by acknowledging the people that did the work. The people in the purple are the people who did the work that I presented today, and of course NIHS and AstraZeneca, with whom the funding would not have been possible for us to do.
Ken, that was really wonderful, and thank you for sharing your unpublished data.